And so we have 64 possibilities of these three-letter words, and they code into something that will allow proteins to be formed with particular linear sequence now of amino acids instead of the ATCG subunits that we find on DNA. What does the code mean? Well, we're still trying to figure out what the code means. You know that the Human Genome Project is more or less completed. We've read through all of the code, but we haven't annotated it. So we have it there, but we don't know what it all means. We don't understand the relationships of some genes. We don't know how genes are controlled in terms of when they're turned on and when they're turned off. Certainly with the, quote, junk DNA, we're really not sure what goes on. But we do have an understanding in terms of the fact that we can identify sequence by using computer programs that look for patterns, and the patterns can pick out genes with really amazing accuracy, and then we're able to, by isolating genes, to look at what individual parts of the code do. All right, again, the question of why so much of this non-essential code. In humans, for example, 95% of our DNA does not code for genes. And of that 95%, what do we see? We see a lot of these repeating sequences that look like they come from ancient viral DNA. So just as viruses today, herpes virus, HIV, can get into our cells and actually become part of our genome, this process has likely occurred for millions and millions of years, and so we have these footprints of ancient traces of virus in our DNA as well. Okay. All right, so one of the things I want to try to leave you with in terms of the new type of evidence for evolution is that if we ask the question, if we say, if evolution is a fact, which we really think it is based on the consilience of evidence from the fossil record, from looking at phylogenetics, morphology, anatomy, physiology, all of these things really lead to the same type of interpretation. Now, the interesting thing about the relationship of these codes of different organisms is that of all of the individual pieces of information, this is probably far and away the most compelling in terms of just knockdown, drag out proof that life is related in a way that is consistent with an evolutionary process. Because to explain it in other ways, to explain how these codes are similar in the ways that they are similar, would take so much hand-waving and tap dancing that it wouldn't be accepted based on basic rules of Occam's razor and parsimony principle. Next, please. Okay, so how do we do the comparison of this biological relatedness? All right, so one of the ways that we used to do it is we used to say, well, if two organisms are related, if we take a protein from an organism and we put it into a bunny rabbit, and that bunny rabbit is going to make antibodies, that antibody is going to bind to the protein that we used to form it. And if we look at the analogous protein from another organism, we can look at how well the antibody recognizes one or the other. I mean, it might seem somewhat crude, but we really learned a lot about evolutionary relatedness, and it was exactly this technique that stunned scientists when they started purifying proteins from humans and chimps. Now, let me tell you that sequencing proteins is much more difficult than sequencing DNA. So the DNA sequencing came long before we got good at sequencing proteins. When we started doing the comparison based on the immunologic relatedness, that's where these figures like humans and chimps are 98% similar came from. And indeed, it's a very useful and powerful piece of information. the next step that came along in terms of comparing these codes was to use another interesting technique. Remember I showed you that the double helix and how it's held together by the hydrogen bonds? Well, one of the things that you can do is you can apply heat, and when you apply heat, you're going to denature those, that double helix. It will come apart. And then what you can do is you can take the pieces of DNA from different species and see what their likelihood is of reannealing. That is, if they're similar enough in sequence, they're going to form back together. And the amount that they reanneal is going to be proportional to the relatedness of the sequence. So that technique is very useful in molecular biology, and the reannealing of the DNA, and you can measure how good the tightness of binding is by asking the question, what temperature do I have to achieve in order to melt them apart? And if it's a really tight, a lot of interaction, then you need a higher temperature. If the annealing is not so good, a lower melting point or melting temperature will work. So both of these techniques, very useful, but they're inferential in a lot of respects. They don't give us, you know, the code, and it's the next piece of information that really was important for that. And so what we finally learned to do is we finally learned to read DNA sequence. We learned how to use supercomputers and very sophisticated techniques. A lot of technology came together, and we were able to basically look at what constitutes the set of instructions for any given organism. And not only are we able to use the technology to read this, but more importantly, we're able to do the comparisons. And the comparisons, the amount of computing power in doing these comparisons is quite simply mind-boggling. But you can use your computer at home and go on to public sites where you can download sequence and you can do comparisons. And if you want to go home and look at particular genes from one organism and another organism and say, well, how closely related do they seem to be based on the actual code, you can do those kinds of comparisons. So it's been really useful, and we've ended up with a lot of good information on that. All of this is based on this principle of parsimony. So when you do these comparisons, basically what you're asking is, what are the hierarchical relationships that you get in terms of degrees of relatedness that are going to require the least amount of coincidence to explain? So if we took hemoglobin from a human and hemoglobin from a kangaroo, and we did sequence the genes, and we say, well, how close are they? Well, they're not terribly close, but we could make up some story about, well, this mutation happens and this happens, and we could say, well, we're really closer to kangaroos than, say, we are to chimps. But if you accept the fact that the best explanation is the most parsimonious explanation, and you look at the human hemoglobin gene and the chimp hemoglobin gene, what you find is that it's really much more similar. You don't have to invoke coincidence. As a matter of fact, you would have to invoke coincidence in the other way to explain, well, they're really similar, but it's just by chance, right? So it's that kind of principle that scientists use in doing these kinds of comparisons. So what we have now is we have the ability to have this phylogenetic tree, and traditionally these phylogenetic trees were largely based on morphology. What do things look like? When you do body plan, when you look at sort of specialized structures, or if you look at physiology, are they warm-blooded, cold-blooded, have hair, do they have feathers? Those are the types of criteria that we used. Now we could basically ignore all of that, look only at the code of these individual organisms, and we end up with a far more convincing and more robust, in the sense of just more data-rich comparisons. And indeed, the other aspect of how this all fits together is it could be a big coincidence, but it seems like these old phylogenetic trees based on morphology, anatomy, and physiology match really quite nicely with the trees that were constructed based on the code, which are totally independent of any of those criteria that you would use to construct this set of trees. So it gets pretty wild if you think that that happens to be by coincidence as well. What's next? This is one of my favorite ways of doing different organisms. I think this is 3,000 different organisms of plants, animals, protists, and fungi, and it says, "You are here." That's where humans are. So this gives us really a remarkable idea. If we want to talk about our planet, if we want to talk about our moral obligations to other animals, to the biosphere, to sustaining the world, all you have to do is look and say, well, we're all related. We share the same code. We came from, long ago, the same places. All of us have common ancestors that at one point would have been a fish, right? So we all come from fish. So, this is really a phenomenal way to look at this history that we have in terms of life. So I'm going to leave you with one last use of this, and this is a practical use of DNA. So we know that we can look at the various relatedness of different species, different groups of animals, but we can also use DNA for practical considerations. We can ask the question, all right, if somebody has a tumor, what's the gene that's gone bad that causes that tumor, and how might we treat that specifically compared to a similar cancer, something called the same, and yet coming from a different genetic cause. So what we're really able to do now is, in many different realms of biology, we're able to ask at the most fundamental level, at the set of instructions, how are organisms related, how are cells related, and when things go wrong in terms of tumors, what's going on in that situation, or in these interesting questions of other sorts of familial relatedness, we can ask these questions as well. So if you would like, and if we have time, I would leave you with a song. Is that okay? So I came prepared. Actually, these are just lying around all over the place. So if this song isn't really politically correct, is that really going to... Or let me say, if any of you are thin-skinned about these sorts of things, you could go get a beer now. I would also say that I didn't write this song, so I can, sort of not take responsibility there. It was actually written by a woman whose name is Dory Previn, and she is the former wife of Andre Previn, who is who's a famous classical pianist and conductor, and she writes some very idiosyncratic songs, and this just happened to be one of my favorite songs that she did, and it's kind of related to genetics. I mean, I don't have a lot of science songs, so I have to sort of, stretch a little bit whenever I can get one that's reasonably close. So. You can sing along too if you want. There's sort of a part that goes like this. Boom, 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 boom. My daddy says I ain't his child. Ain't that something? Ain't that wild? Daddy says I ain't his child. Ain't that something wild? My hair is curly, my freckles are tan. Could my daddy be the garbage man? My legs are stumpy, my fingers are short, like my Uncle Will, who is a bowling sport. My eyes are slanty like Mr. Woo. Hey, Mr. Laundryman, is it you? I'm ugly as John with a big mustache. The mama says poles are a piece of trash. Ooh, hoo, hoo, ooh, hoo, hoo, ooh, hoo, hoo, hoo.